I invite Dr. Amal Johnston from University of Heidelberg in Germany, and we'll be talking on bridging the generation gap on the hunt to solve stress tolerance during vegetative and reproductive development. Dr. Johnston, please.
Thank you, Chairman, and thanks for the organizers for giving me an opportunity to present my lab progress. I apologize, I'm still on sick leave, so I'm trying to stand, looking at you, but I'm not going to turn around. So please just watch the slides. I won't be able to point out with a pointer. So what I'm going to talk to you today, just a bit of concepts, what we are working in the lab, because normally my lab mainly concentrates on reproductive development. By looking at the reproduction, it's a very important phase. So we would be looking at how the male and female gametophyte or germlines contributes to seed filling, imprinting, and so on and so forth. However, while doing that, we decided to look into the sporophytic pathways because it's actually very important to merge both generations. So I would like to convince you that's an important thing to do, not only to look into the sporophyte, but also to look into the reproductive side. So the main model systems that we work on, one is Piscometrella, it's a moss, it's a very lower plant. So if you look into the plant evolution, it's about 500 million years. And if you look into it, the lower plant started off with the water. So if you look in algae, for example, then it goes on to bryophyte. That's an example is a moss. Or look into the later plant that we studied today. So the fantastic talks about all the monocots that you guys talked about and also dicots. So we use Arabidopsis as a very small model system to understand the functional mechanisms. So if you look at the branching point, it's the pteridophytes of fern, and you can see that the watery and less light conditions 500 million years ago, kept going, and these days the dry and abundant light, the land plants started to evolve. So what you see here, there are two halves. So if you look into the dominant gametophyte that predominated in evolution, that got replaced by the dominant sporophyte. Therefore, there is a concomitant gene shift, and regulatory mechanisms are very important to understand how the macroevolution favored the adaptation. So the adaptive evolution of underlying regulatory mechanism is what an important topic in my lab. Sorry, the slide jumped up and down, but I forgot. So the top picture tells you a schematic based on how the somatic development or vegetative development matters, where the embryos start to germinate as seed, and it kept going and flowering and senescence, and that's the whole life cycle. So what you see as a plant, that's exactly what it is. But the forgotten generation, and which should not be ignored if you really want to get higher seed yield, would be the flowers, and not even the flowers, but what is hidden in the flowers, and male and female germline. That is shown in the bottom part of the slide. But I'm not going to go into detail how the gametophyte development, because of very, very minute structures, but would like to only emphasize it's an important phase. So the core pathway that we look into in the lab is the evolutionary ancient RB or retinoblastoma pathway, a very important carcinogenic protein, and it's present in all of us, plants, animals alike. And a single copy PRB, if you mutate, you would get cancer. Textbook knowledge would tell you it's a carcinogenic protein. Not only that, it's a cell cycle protein. It's a repressor. It's a negative regulation of transcription during S phase. Is an important phase to control cell cycle. However, what is really important to know, I'm not going to go into the typical cell cycle thing. It's a phosphoprotein controlled by CDK, cyclin-dependent kinase, and cyclin complexes. But what is really important to know, more than the cell cycle, it has multiple functions. One is female and male germline development or gametophyte development in plants, or sporophytic development, for example, root, leaf, and stomatal cells. They require functional copy of RB. The function can also be cell cycle independent. It's actually an important trait, because if you just simply look at it, it's a very, very essential protein. So if you meddle with it, you must be careful to interpret how exactly you do, and otherwise you just simply end up in cancer-like syndromes. So it's part of a higher order regulatory network. What it is, it's simply a pocket domain protein, so meaning there are two type of pockets called A and B, and they facilitate protein-protein interaction. Cell cycle, it's like a docking station, And you also have a C-terminal domain, which is a typical phosphorylating residues containing domain, or many type of post-transcriptional modifications happen there. And if you look at the right-hand side, you can just see that RBR, or retinoblastoma related, we call it in Arabidopsis, is a single copy. And it seemed to control or associate with higher order regulatory protein, and also the epigenetic factors like DNA methylation controllers, for example, maintenance methylase MET1, or the polycomb complexes, the typical epigenetic complexes, and also trithorax complexes. So that's the known knowledge we have until now. So what we do in the lab is pretty difficult, because we started off with this gametophytic function, because as I said, the protein is very, very important. So if you delete the protein, amorphic I call it, or full loss of function, you would just stop everything. The development doesn't happen. So what we decided to do, we decided to take a hypomorphic approach, meaning taking a couple of residues, and very important residues that could be informative for us along evolution, and try to kill them and see what exactly happens. So this hypomorphic approach, I call this allele RBR4, and this is a mutant that we currently work on, and that gives a quite good indication, because the plant is able to grow and able to have a normal somatic or even a faster development, but only in the germline we have some problem. So just to tell you that initial approaches, like typical transcriptomic approaches, RNA-seq, for example, it told us it's a RB-specific, source-specific response is happening. I mean, a couple of examples may I give you, that there is a shutdown or down-regulation of glucose metabolism, and also there is a lot of effectors of small RNAs being depleted. So there is an epigenetic process is happening, because RB is a higher order regulator. And also, very importantly, what we saw, without giving reference to stress, there's huge cross-regulation or crosstalks between stress and hormone pathways, which is not surprising, being a cell cycle protein. However, many things are cell cycle independent too. So what is happening in female aspect and male aspect, I'm probably going to remove the slide, just wanted to give an introduction that it does have cell proliferation or apomixis-like phenotype. That is not of main importance right for today. But what is exciting for us, that we recently started only a few months ago, and we started to look into the plant, which looked absolutely normal, and looked into what happens if you apply stress. So what we saw on the left-hand side, you can see just the mock and the mutant. In a mock condition, wild type and mutant, there is no difference if you just simply look into it, or if you start to apply the stress. There are an awful lot of changes. For example, the stress amelioration is much better in the RB mutant plants. And this is only just an in vitro assay, and we are doing it much further. And if you do the seed germination assays, you could also see under a salt condition or any type of drought or osmotic stress conditions, you could see that the mutant always has more advantage than the wild type. So we are looking at what kind of transcriptional and metabolic responses. So this is the focus of our work. And again, the RNA-seq says there is activation of quite a lot of stress response genes and hormone genes. And finally, to summarize it, given the time issue, I'm going to summarize very faster. So I would like to convince you that the tool that we have in place, RBR4 hypomorph, is a fantastic tool for us to uncover yet another layer of stress response regulation, may it be osmotic stress, salinity, and much more. So I did not show you all of the things, only a couple of examples. So what is ongoing work, the evolutionary analysis, and what has been lost from Piscometra to Arabidopsis, for example, to compare. And then we try to merge different types of omic data. So we collect the transcriptome data, which is already available, methylome data being conducted, and the metabolome data being conducted. So early indications suggest that RB seem to orchestrate DNA methylation, for instance. Thus, there is a stress imprint, because there is an epigenetic memory. And also there is a dynamic source-sink relationship, which seems to change from sporophytic condition, which I would say like a source, and then when the ovule start to develop like a sink tissue, the whole phenomenon changes. And seem to regulate very nicely all the hormonal pathway which is necessary to ameliorate during stress, and the metabolites as well. So, of course, the ultimate aim is not get stuck with Arabidopsis and Piscometrella, and to transfer the knowledge that we gain in the model system to go to crop plants. With that, I would like to acknowledge all my group members who are shown here. Unfortunately, some people got cut, and one of my PhD student, Jubin, who did bioinformatic work, and acknowledgement to Max Planck Cologne, who helped conducting some large-scale experiment, Bruno, and Professor Grussem, Willy Grussem in Switzerland, my mentor, and Rudiger Hell and Marcus Witz in Heidelberg, and Juan Mateo in Spain, and Professor Hasabe in Japan. And thanks for my funding bodies, and thanks for listening. Thank you.
Thank you very much. And thank you, Dr. Amal Johnston, and thanks to all the speakers for keeping the time.